it would lead to a lot of complications in the genetic information. So to avoid that, nature itself has decided that only one strand, that is that strand which is called as the coding strand. Here transcription always begins in the promoter region and it will terminate or come to an end and stop in the terminator region. Once it reaches the termination, again should it unwind the DNA after that? Not needed, right? So that is why its work is over. Hello everyone, a warm welcome to today's session on chapter 6 of 2nd PUC, that is molecular basis of inheritance. I am Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session of this chapter, we had discussed about a process of replication in DNA, and we studied about Michelson and Stahl's experiment wherein they proved that the replication in DNA is semi-conservative in nature. In today's session, we shall discuss about a very important process in DNA which is called as transcription. So what is that? We shall look into. So talking about transcription, so the word itself says transcription means, in English it means to transcribe something. So you're transcribing means you're copying something. So in this particular case, what is being copied? That is from the DNA, the genes are copied onto the mRNA strand. So transcribing is copying. So therefore, transcription is the process of copying genetic information from one strand of the DNA into the RNA. So here, when I'm talking about RNA, it is the mRNA or the mitochondrial RNA that I'm talking about. So in the case of replication, now again here you have to be very careful about this particular word that I use, from one strand of DNA. Why? Because here both the strands will not participate in the process of replication. Why don't they participate? We shall see in the coming slides. So therefore, only one strand, from one strand the genetic information will be copied onto the mRNA strand, and from this mRNA strand that genetic information will get translated and it will produce protein. So that is the entire mechanism related to transcription as well as translation. But in today's session, we shall concentrate only on transcription mechanism. So the principle of complementarity governs the process of transcription, except that adenosine complements with new forms of base pair, that is with uracil instead of thymine. So what happens here, as we all know that one DNA strand complements the other DNA strand, right? They are non-polar in nature, that is they exhibit polarity, wherein one strand is from the 5' to 3' direction, the other strand is from the 3' to the 5' direction, right? And DNA forms a helical structure. We all know about it. So DNA is a helical structure that looks like this. But when I unwind the DNA and I write two DNA strands and I write the sequence on that, say for example ATGC is the sequence here. So complementary to this, it will be TACG, right? Why is it so? Because we know that adenine base pairs with thymine and guanine base pairs with cytosine, right? So that is why. This is in the case of when both the strands are DNA. But what about one strand is DNA and the other strand is mRNA? So when one strand is DNA and other strand is mRNA, the mRNA strand will be formed to this particular strand of DNA, which is read from 5' to 3' direction, wherein in the place of thymine, adenine will be there. In the place of adenine, will thymine be there? No. Why? Because RNAs do not have thymine. Instead, they have uracil. Therefore, it will be U, then this as such G and C. So that is how the base pairing occurs. So therefore, the mRNA strand that is formed for one of the strands of the DNA will complement it. It is based on complementarity principle that was given by Watson and Crick. And not just that, only one change that we find here is the adenine will base pair with uracil and not with thymine, as in the case of DNA. So that is the only difference that is found here. So however, unlike the process of replication, which once set up, the total DNA of the organism participates in the process of replication, wherein the total DNA of the organism gets duplicated, right? But in the case of transcription, the total DNA will not get duplicated. If you remember, or you can go back and watch the previous session of this particular chapter wherein we had learnt about replication, right? In that I had mentioned that in replication the strands will unwind, and those two strands, which are called as the template strand or the parental strand, they will participate in the process of replication, wherein to each of these template strands the daughter strands will be formed, wherein therefore two DNAs will be formed. But in the case of transcription, the main difference is that here also the unwinding of the DNA will take place. Just like how for replication the unwinding of DNA would occur, here also unwinding of DNA will occur. But will both the strands participate in transcription? No, it is only one strand that will participate in transcription. Which strand? That strand which reads from 5' to 3' direction. So that is the only difference between replication and in the case of transcription. And in replication, one more daughter DNA strands are formed, but here DNA strand will not get formed, but instead a mRNA strand will get formed for one of the DNA strands. So that is the difference between replication and transcription. So here the question arises now. When I was telling you about the definition for transcription, I told you transcription is a mechanism wherein to one of the DNA strand a complementary mRNA strand is produced. Now to one of the DNA strand, why does only one DNA strand participate in the process of replication? Two strands could have participated, right? Because for two strands also mRNA can be produced, complementary mRNA strand. Why only one strand participates? There are two important points that has to be kept in mind. We shall look into it. So why both the strands are not copied during the process of transcription, whereas in replication both strands were participating in replication, but in transcription only one strand is participating. Why is it so? So first, if both strands act as a template, they would code for RNA molecule with different sequences, and in turn they would code for proteins, and the sequence of amino acids in the proteins would be different. So that is why only one segment of the DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. So what does this mean? So here, as I told you, DNA is double stranded, right? So in the DNA we have two strands, one strand reading from 3' to 5' direction. Because they exhibit polarity, the other strand will read from 5' to 3' direction. So here I'll write the sequence for this. For example, ATGC is the sequence, and obviously the other strand will have a sequence complementing the strand above, right? So what will be the sequence here? TACG. Now what might happen if both the strands participate in the transcription process? Say for example, here one mRNA is formed. When the mRNA is being formed, what nucleotides get added? The nucleotides which are complementary to this particular strand of DNA, which is reading from 3' to 5' direction, that will get added, right? So therefore here it will be U. It is U, it's not T. Again, why? Because I'm talking about the mRNA strand. Here I'm not talking about the DNA strand. So therefore here it will be U, this will be A, this is C and G. Next, one more strand here, it will be A, U, G, C. Now, here if you see the triplet codon, it is UAC, here it is AUG. Now, it is a confusion for a body to understand what proteins has to be produced, because too many number of proteins would have produced, because these would code for different amino acids. So that is the reason. It would lead to a lot of complications in the genetic information. So to avoid that, nature itself has decided that only one strand, that is that strand which is called as the coding strand. This is called as the template strand, which will not participate in replication. This one, The strand that is reading from 5' to 3' direction is called as the coding strand, and only that will participate in the transcription process here. So next point, why only one strand participates in the transcription process? So second point is that the two RNA molecules, if produced simultaneously, they would be complementary to each other. Hence, they would form a double strand. Now here, DNA, they are double-stranded. They form a helical structure. Why? Because they are complementary to each other, and that polarity makes them to form a helical structure, that is, they will coil up. Now again here, say for example, I'll draw the two DNA strands. One is from 3' to 5' direction. The other one is from 5' to the 3' direction. So I'll write the sequence ATGC. Here complementary to that, it will be TACG. Now a mRNA strand that is formed for this, it will have a sequence. So this is the mRNA strand that is formed. If it gets formed, actually here mRNA strand will not get formed. If it gets formed, what will happen? I'll just show you. So here it will be A, then we have C and we have G. Next here again we have A, U, G, C. Now if you see here, U is complementary to A, right? A is complementary to U, C for G, G for C. So because both these mRNA strands, these two mRNA strands that are there, because they are complementary to each other, they might start to coil and they themselves might form a helical structure. And if they again form a helical structure, from where will the proteins get produced? It will not get produced, right? At the end of translation, proteins will, or translation mechanism will not at all occur. So that is why, in order to prevent all those problems, nature itself has decided that the DNA, only one DNA strand will participate in the transcription process, and it is not two strands. So that is the reason why both the strands of DNA are not copied, or two DNA strands, two mRNA strands are not produced. So now we know the definition of transcription. We know why only one strand. Because after all this, we came to know that only one strand participates in the transcription. Which strand? The strand reading from 5' to 3'. Now we know a little about it, right? So let us move on to the actual mechanism of transcription, wherein we'll first learn about the transcription unit. So what is a transcription unit? A transcription unit is nothing but a stretch of DNA strand which actually participates in the transcription. Again here, you need to understand that the entire DNA strand will not participate in the transcription. It is only from certain region, from certain sequence to a certain sequence, the DNA strand participates in the transcription. So therefore, that stretch of sequence is divided into three main regions. One is a promoter region, the structural gene, and a terminator region. So these we shall study about these one by one. What is their function, what do they do, and all that. So you can see here we have the promoter region, then comes the structural genes, and at the end we have the terminator region. What are they important for? We shall look into, and the transcription will take place only from here up till the terminator. It will not begins anywhere else in on the DNA strand. So moving on to the transcription unit, since the two strands have opposite polarity and the DNA dependent RNA polymerase enzyme. Now you need to remember this enzyme, which is DNA dependent RNA polymerase enzyme. If you remember, in the session wherein we had dealt about replication, we had learnt about an enzyme which was DNA dependent DNA polymerase enzyme. Now there it was called DNA dependent DNA polymerase enzyme. Why? Because depending on one of the strands of the DNA, another DNA strand was getting formed there. So that is why it is a DNA dependent DNA polymerase enzyme. Now in the case of transcription, for one of the strands of the DNA, will a DNA get formed? No. What will get formed here? It is a mRNA that is getting formed, right? So that is why this particular enzyme, it makes use of one of the DNA strand to add complementary nucleotides, therefore forming a mRNA strand. Hence, it is called as a DNA dependent RNA polymerase enzyme. So remember this enzyme, it's very important in the next discussions. So this enzyme, it also catalyzes the polymerization in only one direction, that is from 5' to 3' direction. So therefore, which is the strand that participates in the transcription? It is that DNA strand that can be read from the 5' to 3' direction. Only that will participate in the process of transcription. And it has, that strand has one more strand which is showing polarity, that is 3' to 5', and that strand acts as a template. Therefore, the strand which is read from 3' to 5' direction is called as the template strand, and the strand that is read from 5' to 3' direction is called as the coding strand. So therefore, it is this particular coding strand that participates in the transcription process, whereas the template strand will not participate in the transcription process, meaning mRNA gets produced only for the coding strand and not for the template strand. You have to keep that in mind, and therefore this one is referred to as the template strand, and the other strand which has the polarity 5' to 3' direction and the sequence same as RNA, thymine at the place of uracil is displaced during transcription, meaning for this particular strand the mRNA molecule will be formed, wherein this mRNA molecule will have adenine, uracil. There is no thymine here because it is a mRNA. Then it will be guanine, cytosine, then adenine, uracil, guanine, cytosine. Like this it will go on adding. So if this particular complementary nucleotides has to get added, which enzyme will help? It is the DNA dependent RNA polymerase enzyme that will help. So now what all points we came to know? We came to know that only one strand participates in the process of replication. Which is that strand? That strand is called as the coding strand, which is read from 5' to 3' direction, and to that complementary mRNA strand gets formed, wherein the polymerization of the mRNA strand is helped by an enzyme which is called as DNA dependent RNA polymerase enzyme. So much we know them. And also we know that here thymine, in the case of mRNA, thymine will be replaced by uracil. And strangely, this strand, and one more thing, there is a fact here, they have named this strand as coding strand. But that strand actually doesn't participate in the coding of any of the genes. But still, because they help, this strand participates in the formation of mRNA strand, which takes part in the coding of the genes. Therefore, this strand of DNA is called as the coding strand. So that is one information that we need to keep in mind. So don't get confused. These genes that you see here, they do not code for any of the proteins. But since they participate in the formation of mRNA strand, which codes for proteins, it is called as the coding strand. So therefore, coding strand is the one that helps, that involves itself in the formation of the mRNA strand. So this is about the transcription unit. So moving further, we shall study about the promoter region, the terminator region, the structural gene and all that. So the promoter and the terminator flank the structural gene in the transcription unit. That's true, right? They flank. They are present on opposite sides of the structural gene. The terminator and promoter present on opposite ends of the structural genes. Therefore, it is said that the promoter and the terminator region flanks the ends of the structural genes. And the promoter is said to be located towards 5' end. If you can see here, promoter. Don't consider this strand. Why I am not considering this strand? Because this strand is 3' to 5' strand is a template strand which will not participate in replication, which will not participate in transcription. Which one will participate in transcription? It is only the coding strand that participates in transcription. So therefore, I have to consider these two ends, 5' end and 3' end here. Don't forget about the template strand. That is not at all needed here. So we'll just forget about it. So here, where is the promoter present? It is present in the upstream, right? Upstream. Why it is upstream? Because it is present before the beginning of the structural genes. So therefore, it is present upstream towards the 5' end. Whereas you can see it is present downstream because it is present after the structural gene. It is present downstream towards the 3' end. So therefore here replication always begins in the promoter region. So here transcription always begins in the promoter region and it will terminate or come to an end and stop in the terminator region. So that is why the entire strand of DNA will not participate in the transcription process. It is only the transcription occurs from the promoter region up till the terminator region. Only till that transcription will occur. So this is about the transcription unit. So moving further, so by switching its position with terminator, the transcription can be reversed, that is in some of the organisms, and the terminator is usually, as I told you, it is located towards the 3' end, that is towards the downstream. So all that I have explained it to you. So moving further, we shall study about the transcription unit and the gene. So we shall study about the transcription unit and the gene. So what is a gene? So gene is nothing but the functional unit of inheritance. Why it is the functional unit of inheritance? Because it is carried from the parent to the offspring. Therefore, it is the functional unit of inheritance, right? Because we inherit or we get the characters from our parents. How do we get the characters from our parents? It is because of the transfer of the gene. So that is why. So therefore, gene is the functional unit of inheritance and the DNA sequence coding for tRNA or RNA molecule also define a gene. So in the tRNA or the RNA molecule that is also participating in the coding sequence, they are also a functional unit of the, or they are also defined as a gene. So next talking about a cistron. What is a cistron? Genes are usually in the form of triplets, AUG, ATG, ACG, like that. Okay. Now what is a cistron? Cistron is a segment of DNA. So a segment of DNA coding for a polypeptide is called as a cistron. So the structural gene in a transcription unit could be monosystronic in the case of eukaryotes and it could be polysystronic in the case of bacteria or in the case of prokaryotes. So therefore, remember the functional unit of inheritance is a gene. The segment of DNA that participates in the coding process is the cistron. The cistron in the case of eukaryotes is monosystronic and the cistron in the case of prokaryotes is polysystronic. So moving further, let us study about the monosystronic structural genes. So in monosystronic structural genes are presented. So we learnt about the promoter and terminator region and all that. Now we are studying about that particular set of genes that are present at the, or the strand of DNA at the center between the promoter and the terminator that participates in the transcription process. That structural gene is of two types. In eukaryotes, that segment, it is called as monosystronic. In prokaryotes, it is called as polysystronic. Now we'll study about monosystronic structural gene. So in eukaryotes, the monosystronic structural genes, they have interrupted coding sequences. Therefore, the genes are split. Say for example, they'll have a coding strand and a non-coding strand. A coding strand and a non-coding strand. So, the coding sequences are called as exons and these are called as the introns. So, therefore, these introns, which are the non-coding sequences, they do not participate in the production of proteins. So, therefore, these has to be removed and only the exon should be retained. So, because eukaryotes, the mRNA strand that is formed will have both the coding sequences and the non-coding sequences when it is a nascent mRNA or immature mRNA. Therefore, it is said that the structural genes in the case of that is in the case of eukaryotes, it is monosystronic in nature. So, the coding sequences are expressed as exons. They are called as exons. As you can see here, these are the exons, introns, non-coding, exons, coding. So that is how the eukaryotic DNA strand will look like. Because they have coding and non-coding sequences, even the mRNA strand that is formed complementary to it will also have a coding and a non-coding sequence there. That is, they'll have a exon and an intron. Remember, exon is the coding sequence, intron is the non-coding sequence. So moving further, as I told you, what are exons? Exons are said to be those sequences that appear in a mature or processed RNA. That is true, right? Because I told you in the immature mRNA or the nascent mRNA, the exons and introns both will be present. Those introns will get removed. The exons will all join together and they will form a mature mRNA wherein only exons will be present. That is, the entire stretch of RNA will now become a coding strand. So that is about it. And introns or intervening sequences do not appear in a mature processed RNA. Same thing as I had explained to you earlier. Knowing about the types of RNA, because in transcription, 3 types of RNA participate. That is why we need to know about those 3 types of RNA. So the types of RNA in bacteria, that is in the case of prokaryotes, there are 3 major types of RNA that participates in the transcription process, and all 3 RNAs are needed in order to synthesize a protein in a cell. So what are those 3 RNAs? We shall look into. First one is the messenger RNA, that is the mRNA, and the mRNA, it provides the template. It acts as a template for the tRNA and the rRNA. What is this tRNA? It is the transfer RNA because it is the one that carries the amino acids to the mRNA. So that is why it is called as a transfer RNA, which transfers the amino acids to the mRNA, therefore helping in reading the genetic code. So next is rRNA. It is ribosomal RNA. Ribosomal RNA has a very important function, that is structural and catalytic role during translation, because that is the one that helps in translating the amino acids into a protein, and that is how proteins get produced in a body. So therefore, messenger RNA, the ribosomal RNA and the tRNA participates in the process of transcription in the case of bacteria or in the case of prokaryotes. Usually, E. coli is taken as an example to explain this transcription translation mechanism. So, usually E. coli, which is a bacteria, which is a prokaryote, is usually taken as an example to explain this transcription and translation. So, now because we know about transcription, the types of RNAs that participate in the transcription, and we know about the transcription unit and all that, it will be easy for us to understand. And we know about the enzyme that participates in transcription, all that because we know, and which strands participates in transcription, that is the coding strand, right? Because we know all that, it will be easy for us to understand the process of transcription. So let us study about the process of transcription. In that, first let us concentrate on studying about the enzymes and transcription. So which is the enzyme? Earlier also we came to know about it. It is the DNA dependent RNA polymerase enzyme, right? So what does this RNA polymerase enzyme do? So the RNA polymerase enzyme, it will go and bind to the DNA. Again here, there are 3 transcription units in the DNA. What are those? Promoter region, structural genes and the terminator region, right? Now, where will this particular RNA polymerase go and bind? Will it bind anywhere it likes on the DNA strand? No. Why? Because I told you transcription will take place only from the promoter region and it will end till the terminator region, right? So, therefore, here first it will go and bind to the promoter region. Promoter region is the one that initiates the process of transcription. So, if you can see here, RNA polymerase goes and binds to the promoter region, which is present upstream towards the 5' end of the coding strand of the DNA. Now it binds to the promoter and it will initiate the transcription. It will begin the transcription. So for initiation of transcription, what is needed? The binding of the RNA polymerase enzyme to the promoter region is very, very important. And apart from that, this particular RNA polymerase enzyme, it makes use of nucleoside triphosphates. Why it makes use of nucleoside triphosphates? Because as and when the unwinding of DNA occurs from the promoter region, the mRNA strand will start to form. If that mRNA strand has to form, then to the mRNA strand, the complementary nucleotides has to get added. If the complementary nucleotides has to get added, then nucleoside triphosphates are very, very important there. So that is why RNA polymerase makes use of nucleoside triphosphate as a substrate, and it will polymerize in a template dependent fashion following the rule of complementarity. Again, the same thing. You know the rule of complementarity, wherein a base pairs with uracil, because it is the RNA strand that is forming here, thymine with adenine, guanine, cytosine, guanine, like that. So therefore, that is what actually happens. So that occurs in the process called as initiation. So for initiation to occur, RNA polymerase enzymes binding to the promoter region is very, very important. And if the formation of the mRNA strand has to occur, then along with that, the making use of nucleoside triphosphates are also very, very important. And it also facilitates the opening of the helix and continuous elongation. So I told you, this RNA, it will go on opening the helix. See here, it opened the helix, formed the mRNA strand. Once the helix gets open, it will close back. And again, wherever the RNA polymerase is moving, there and all, the helix, the DNA helix will start to open, and along with that, the RNA polymerase will make use of the nucleoside triphosphate to add RNA molecule, mRNA molecule. And the mRNA molecule, till where it will get formed? It will get formed until the RNA polymerase reaches the terminator region. And that is why, because the elongation or the addition of nucleosides to the mRNA, complementary nucleotides to the mRNA, takes place continuously up till the terminator region, this particular step is called as elongation. And termination means at the end, what will happen, we shall look into. So therefore, transcription takes place in 3 steps. One is initiation. In the initiation step, the RNA polymerase will go and bind to the promoter region. It will unwind the DNA strand. Once it unwinds the DNA strand, it will start to make the mRNA strand. For that, it will take the help of nucleoside triphosphate, and next elongation will occur as and when the RNA polymerase moves on the DNA strand. The DNA strands will unwind, the coding strand will participate in the formation of the mRNA. The mRNA strand will start to elongate, making use of the nucleoside triphosphates, and therefore that process called as elongation. Up till when the elongation will take place? Up till it reaches the termination region. Once it reaches the termination region, what will happen? We shall see. So once the polymerase reaches the terminator region, the nascent RNA or the immature RNA falls off, so also the RNA polymerase. This results in termination of transcription. So as you can see here, once it reaches the termination, in the step termination, the RNA, nascent RNA or so this is the nascent RNA, it will just fall off. Along with that, RNA polymerase will also come out. Why RNA polymerase will come out? Once it reaches the termination, again should it unwind the DNA after that? Not needed, right? So that is why its work is over. So that is why it will just come out of the DNA strand. Now this nascent RNA, it is an immature strand. This has to become mature. In the case of eukaryotes, in prokaryotes it is fine because they will not have exons and introns. They will have only exons there. But in the case of eukaryotes, they will have to undergo various other processing steps, because this immature mRNA will have both the introns and the exons, that is the non-coding and the coding sequences. The non-coding has to be removed and the coding sequences only has to be retained. So therefore, they'll move into the next step. What happens? We shall look into. So we shall study about the process of transcription in bacteria. So in bacteria, since the mRNA, as I told you, since the mRNA does not require any processing to become active. Remember, in bacteria, the mRNA does not require any processing to become active. Why? Without any processing, directly the mRNA will enter into translation process. Why? As I told you, because in the case of bacteria, the mRNA strands will not have, bacteria as a prokaryotes. these mRNA strands will not have on their sequence, they'll not have introns, only exons will be present. So therefore, there is no need for entering into a step wherein introns has to be removed, exons has to be retained, and all that step is not needed. Therefore, they'll directly enter into translation mechanism. But in the case of eukaryotes, this process is needed. So what happens there? We shall look into. So we shall study about the process of transcription in eukaryotes. All the steps until termination was fine. But that nascent RNA, that mRNA that comes out, right, in the case of eukaryotes, it will have introns as well as exons. Therefore, introns should be removed, only exons should be retained. How it will occur? We shall look into. So therefore, the eukaryotic transcription mechanism and translation mechanism is a bit complicated when compared to that of prokaryotes. So here there are at least 3 RNA polymerases enzyme that participate in the process of transcription in the case of eukaryotes. So in prokaryotes we had studied that only one transcription enzyme participates, right? That was RNA polymerase enzyme. But in the case of eukaryotes, 3 different RNA polymerases enzyme participate in the process of transcription. So the RNA polymerase 1, so it is RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. Now here itself you need to understand the difference. Prokaryotes, only RNA polymerase participated. Eukaryotes, RNA polymerase 1, 2 and 3 participates in the process of transcription. So the RNA polymerase 1 transcribes the rRNA, that is the ribosomal RNA, which are made up of 28 subunits, 18 subunits and 5.8 subunits, whereas the RNA polymerase 3 is responsible for transcription of tRNA and 5 subunit ribosomal RNA and also some small nuclear RNAs as well. But for us now, the tRNA is the one that is important, because in eukaryotes we learnt that 3 types of RNAs participate in transcription, right? What are they? They are the mRNAs, they are the tRNAs and they are the rRNAs, right? So, RNA polymerase 1 transcribes the rRNA, RNA polymerase 3 transcribes the tRNA and RNA polymerase 2 transcribes the precursor of mRNA. That is, instead of mRNA, it will also help in transcribing a particular RNA, which is called as heterogeneous nuclear RNA or the hnRNA. Now moving further, the second complexity is that the primary transcript will contain both the exons and the introns, which are non-functional. Exons are functional, introns are non-functional. Exons are coding, introns are non-coding. So therefore, as you can see here, this is about the process of transcription in the case of eukaryotes. As you can see, they are made up of exons and introns, exons, introns, exons and introns. Now, once the nascent mRNA after termination comes out of the DNA strand, it will contain, in the case of eukaryotes, it will contain both exons and introns, that is the coding and the non-coding sequences. So now this particular exon and intron should be removed. So if that has to be removed, capping has to occur. What happens there? We shall look into. Hence it is first splicing should occur. Hence it is subjected to a process called as splicing. Splicing means to cut and to remove something, wherein introns are removed and exons are joined in a definite order, and the heterogeneous RNA undergoes additional processing called as capping and tailing. So now you understood, right, what is heterogeneous RNA, because RNA polymerase 3 helps in the formation of a heterogeneous RNA. Why it is heterogeneous? Because it is having 2 types of genes. One is coding, which is coding, the other one is non-coding. So this heterogeneous mRNA should become a mRNA having only coding strands. So for that, splicing has to occur. If splicing has to occur, what should happen means exons should be retained, introns will be removed. Exons will be retained, introns will be removed, and when they are removed, what happens here is once the introns are removed, gaps will be there between the exons, right? So therefore, the exons should be joined. They will be joined with the help of ligases enzyme. They will be joined, and before the process of splicing, there is one more process that will occur, which is called as capping and tailing. What happens in capping? In capping, actually, this methionine guanine triphosphate will form a cap at the 5' end, and apart from that, a poly A tail will be formed. In the 3' end, there is polyadenylation, means addition of a long chain of adenine group will take place. So, so much will occur. What will occur? Capping will occur, wherein methionine guanine triphosphate will form a cap at the 5' end of the nascent mRNA. After it forms a cap, what happens? RNA splicing will occur, wherein all the exons will be retained, introns will be removed. Right? Exons will be retained, introns removed. So that is how it occurs. So once now the gap that is there, that has to be filled, and now if you can see here at the end, only the exon will be retained here. That is the coding strand will be retained. Now this messenger RNA is a mature mRNA. Mature. So mRNA is a messenger RNA. Sorry, it's not a mitochondrial RNA. It is a messenger RNA. So this messenger RNA, which is mRNA, will become a matured mRNA. So therefore, apart from that, after capping and after splicing process and all that, there is also a process called as polyadenylation will occur. Polyadenylation means poly means many. Adenylation means addition of adenine group. So in the 3' tail of the, that is the mRNA strand, the addition of a poly, that is adenine group will take place. Therefore, it is called as a poly A tail. So a tail will be formed which consists of only of adenine. So it is called as a poly A tail. So much, and later on what will happen? Now the mRNA strand that is there, it will have only the poly A tail. It will have the methionine guanine triphosphate capping and only the exons. Now it is a mature mRNA, and that mature mRNA will participate in the translation process. So in capping, an unusual nucleotide called as methyl guanosine triphosphate is added to the 5' end of the heterogeneous RNA. Why it is a heterogeneous RNA? I told you, because it consists of two types of sequences, coding and non-coding sequences there. And in tailing, adenylate residues or adenine will get added. How many? 200 to 300 adenine will get added, as I told you earlier, and they are added at the 3' end in a template independent manner, and this fully processed hRNA is now called as the mRNA. So that's the main difference between prokaryotes and eukaryotes. Prokaryotes, they will not have a heterogeneous RNA. It is directly mRNA. Why not a heterogeneous RNA? Because there, that RNA that is formed will not have a introns and a exon. Only exons will be present. But in the case of eukaryotes, what happens? It is a heterogeneous RNA, because heterogeneous means two different sequences are present here. That is coding sequence and non-coding sequences, wherein the coding sequences will be retained, non-coding will be removed. Therefore, a mature mRNA will be formed. That mature mRNA will come out of the nucleus, it will enter into the cytoplasm, and it will participate in the translation mechanism. This translation mechanism we shall study in the next coming sessions. So this was about this particular session, wherein we completely studied about transcription. We understood the differences between the transcription mechanism in the case of prokaryotes and eukaryotes. It is quite simple. The first, everything is same. Only thing is, once the mRNA is formed, there is a difference in the eukaryotic transcription mechanism. That's the only difference. So that was about today's session. In the coming session, let us discuss about the genetic code and the mutations and the genetic code. In detail, we shall study about the genetic code. So we'll meet again in the coming session. Thank you.